Hello and welcome. Today we will be talking about viral infections. And our purpose for today is to make sure you can compare the structures of viruses to cells, how do viruses reproduce, and what is the role of viruses in causing human diseases. There are also two different cycles, so we're going to make sure you can compare and contrast the lytic and lysogenic cycles. Just real quick, some of the reasons we should care about this, the diseases that these can cause range from mild to life-threatening, and there are all sorts of things that we have to consider when thinking about viruses. So this is why we should care and why we should learn about viruses. So one of the things to understand is that viruses are not truly considered living organisms. Unlike cells, viruses cannot reproduce on their own. They require a host cell to make more viruses. Viruses do not grow and develop. They do not carry out any cellular processes like photosynthesis or cellular respiration, and they do not have the ability to maintain homeostasis like we've talked about in cells. However, viruses do contain genetic material. This means that viruses can evolve. These are two of the characteristics of living things that we did talk about. Viruses are incredibly small compared to the smallest prokaryotic cells. You can see the smallest prokaryotic cells is in this range, and then viruses are way down here, measured in nanometers, not micrometers. There is a lot of diversity with this group. Not all viruses are the same. And the symptoms and damage caused by viruses often depends on what type of cell the virus is infecting. So some viruses are pretty specific about what type of cell they will infect. For example, HIV infects helper T cells of the immune system. That's it. That's what they infect. And then this weakens the immune system of the host and can lead to more infections and illnesses for the host. So the damage caused by the virus is actually because it's weakening the immune system. And viruses will affect different cells depending on the type of virus. And while there are several differences between all the different viruses, they all have a basic structure. There's the genome surrounded by a capsid. The genome is either going to be DNA or RNA, that's the nucleic acid, and it contains the instructions for making new viral capsids. The capsid, which you can see right here in the picture, is a protein coat that protects the genome. The DNA or the RNA is what is used to infect host cells, and the DNA is used by the host cell's organelles to make new virus parts through protein synthesis. Some viruses will also contain this outer envelope that you see right here, and this can serve as a receptor for easier attachment to host cells. There are many types of viruses. Any type of virus any type of cell can be infected by certain viruses. This means that any type of living cell can be infected by a virus. Viral infections do not just affect animals. Don't worry though, plant viruses don't make us sick. They make plants sick. All viral infections work in pretty much the same way. Some slight differences, but pretty much the same way. It starts when the virus attaches to the host cell using the envelope proteins. The envelope proteins that you can see here on the virus have a specific shape, and it matches the receptors on the cell membrane of the host cell. The viral DNA, or if it's an RNA virus, there's an extra step right here that we're going to skip for just to make it simple, but the viral DNA is going to enter the cell. When it enters the cell, it goes to the nucleus where the host cell's DNA is. The cell is not going to recognize this DNA as an invader because DNA is universal. There aren't any major differences that would tip off the host and make it go, hey, wait a second, you don't belong here. Once the DNA is in the nucleus, it will integrate with the host cell's DNA. From here, the DNA is replicated just like replication happens in all cells, not just eukaryotic cells, in all cells. Then the DNA is used to produce through protein synthesis is used to produce more viral structures like the capsids. The new viruses are assembled and then released from the cell. This causes the cell to lyse or break open, which can kill the cell. The new viruses can then infect nearby cells, and this can lead to tissue damage in the person because of all those cells being lysed. The disease or illness that results from this damage, is what results from this damage. Not all viruses lyse cells. Some will disrupt homeostasis. Some can cause the immune system to destroy the host cell, and then others actually lead to cancer. For example, I've heard it papillomavirus and papillomavirus, so you might have heard it two different ways. This virus can actually lead to cervical cancer in females. When we're looking at viral infections, the first type of infection to discuss is called the lytic cycle. The lytic cycle is the shorter cycle and is a period when the virus is active. During the lytic cycle, the virus is actively replicating and making new copies. The viruses will lyse the host cells. To lyse means to break open. So the virus lyses the host cell and causes a person to experience the symptoms of the illness. So in this picture, what we see is the lytic cycle. Some viruses only go through the lytic cycle, and when the host cell is infected, the viruses will continue to infect new cells and cause damage until the immune system can actually contain the infection. Influenza, or flu, is an example of a lytic virus. You see we have stage 1 is attachment, attaches to the host cell. Step 2 is penetration, the viral DNA enters the host cell. Biosynthesis is when the virus is using the host cell's machinery to make new viruses. It replicates the DNA, it makes new proteins through protein synthesis on the ribosomes, and then other organelles actually assemble this whole thing and package it just like it would cell products, and that happens during maturation. And then lysis is when the viruses break out of the cell and releases these new viruses. And this one right here, this bacteriophage, this actually infects viruses. This is a bacterial virus, not a human virus. The other type of infection is the lysogenic cycle. This is the longer cycle. See how it has the longer word? Longer cycle. During the lysogenic cycle, the viral DNA is incorporated into the host cell's genome, but it's not active. This is known as a period of latency. The cell will continue to grow, replicate its DNA, and divide into new cells. The viral DNA is copied into the new daughter cell along with the host cell's DNA. The viral DNA can actually remain dormant, that's that latency period, for years before becoming active. And scientists aren't really sure what causes the viruses to become active, although the environment does seem to play a significant role in this. When the DNA becomes active, the virus enters the lytic cycle and begins destroying cells. Examples of lysogenic viruses include HIV, chickenpox, and even herpes. So you've probably heard of people who had chickenpox when they were young. It remains dormant in bundles of nerve cells for years. And when it becomes active again, years later, sometimes in their 50s or 60s, it can cause shingles. And this seems to be like stress or other illnesses, so the environment of the person causes this virus to become active again. For treatments for viruses, it's important to understand that since viruses are not living cells, antibiotics or drugs designed to kill cells will not work on a viral infection. However, we can protect ourselves from getting an infection by using some common disinfecting cleaners on surfaces and practicing good hygiene, such as washing our hands. Household cleaners and soap help to break down that protein code and cause the virus to become inactive. Some viruses will not survive drying out, so they can't hang out on surfaces or in the air for very long. And so that's why, kind of isolating yourself when you're sick helps to prevent the spread. Scientists have successfully developed some antiviral drugs that are helping to combat the spread of things like HIV, herpes, and hepatitis, and even influenza. However, it's important to understand that there's no cure for most viruses. What doctors focus on is treating the symptoms and controlling those symptoms to keep the person comfortable and keep the body so it can survive and mount a defense using the immune system. And the immune system will control that infection. Vaccines, we've all heard of vaccines, and what vaccines do is they introduce small amounts of inactive virus or parts of the virus known as antigens to create an immune response in the body. And the first vaccine was developed by Edward Jenner to inoculate or introduce small amounts of the virus into a healthy person's body, like people against smallpox. The body produces antibodies in response to the antigens in the vaccine. This helps the body protect against future infections. If the virus is introduced into the body again, the immune system is ready to defend it again, and most people do not get sick a second time. And if they do, it's usually the symptoms are also milder. There are exceptions to this, but it's generally what happens. Viruses have been used a great deal to reduce the number of illnesses or death due to viruses. And diseases that were just a way of life for older generations, such as polio, measles, mumps, and rubella, are actually quite rare. And people like me who didn't receive the chickenpox vaccine and had chickenpox, I have to worry about getting shingles later in life. But now children are usually vaccinated and protected from both. And it's thanks to vaccines that things like smallpox have been eradicated from nature since 1979. Scientists are hoping they can do this with other diseases as well. One of the problems that scientists run into is, like cells, when DNA is replicated, mistakes happen. These mistakes, or mutations, are a source of genetic variation, which can lead to a changing population of viruses. This means that viruses can evolve or change over time. Let's say the DNA of a pentagon virus may replicate incorrectly, resulting in a different or triangle virus. We're not really changing the shape of the virus like this. It's just kind of a model to show you what would happen when the DNA changes. When the triangle virus is produced, the DNA may cause different outer proteins to be made, and the result is this triangle virus is not recognized by the body as being the same as the original invader. So the body doesn't have antibodies already made for that. This can result in a wide range of strains or shapes that exist in the human population. And viruses, as they continue to mutate, it gets harder for the body, and as they continue to mutate within the host, this is going to cause problems for scientists, but not all viruses mutate at the same rate. This evolution can make it hard for scientists when trying to develop a vaccine against the virus, because there's so many strains out there. A vaccine effective against one strain may not be effective against other strains. And we see this in influenza vaccines. Scientists make vaccines for 3 or 4 common strains, but that may not be the only strains that a person comes in contact with. The result, the person gets the flu anyway, not from the vaccine, but because the different strain means that we didn't have antibodies against it. This also causes problems for our own immune system, because you might get the flu and you build up an immunity to that strain, but then you come in contact with a different strain and you get a different form of flu, but you still feel sick either way. And the last thing I want to talk about is how viruses are being used in medicine. Since viruses can carry the DNA straight to the nucleus of the cells that they infect, scientists are working on treating gene defects in cells. The virus delivers a working gene to replace a faulty gene. It's new technology, but scientists are hopeful that the treatments will be successful. Scientists are also working to make bacteriophages that can infect bacteria that are resistant to antibiotics as an alternative to antibiotics. But this does have some problems and a little bit of drawbacks on this that they're working through. Scientists are also hoping that they can use oncolytic viruses, these are the ones that infect cancer cells, to treat cancer in the future. Here are a few follow-up questions I'm going to leave you with. Some things for you to consider as you move forward thinking about viruses and learning about viruses.